Hi, good morning, everybody. Thanks for being here today. My name is Jeremy Waddell. I work for the USC Wetland Team, and I'm here today to introduce our speaker for this week's edition of Watershed Wednesdays. We still have some more people popping in. Thanks again for being here. Watershed Wednesdays are hosted by the Upper Susquehanna Coalition and the Otsego County Conservation Association, with our final webinar of the session is scheduled for next Wednesday, November 20th. The sessions are recorded and can be assessed both on the USC website and YouTube page. During the presentation, if you have any questions, just drop them in the chat and we will read them at the end of the presentation. To get things started, our presenter today is Brian Hefferon. Brian is a Master of Biology student at SUNY Oneonta and is set to graduate this December. As part of his degree program, Brian has studied freshwater mussels in the Upper Susquehanna and Mohawk River watersheds for the past 3.5 years. Brian is also a full-time employee of the Fisheries, Wildlife, and Environmental Sciences Department at SUNY Cobleskill, where he provides instructional support to the fisheries program. During his graduate research with SUNY Oneonta, Brian has built a propagation facility at SUNY Cobleskill, where he studies and propagates freshwater mussels. Thanks again for being here today, Brian. I'll turn it over to you.
Yep. Thanks, Jeremy, and thanks everybody for joining. So today I'm really just covering one of my couple chapters for my thesis research, right? So for my research, I've been exploring yellow lamp mussel life history. And this group, why are we interested in yellow lamp mussel? And really that's because yellow lamp mussel is one of three really high priority species in the upper Susquehanna River, right? So we also have green floater, which is a state threatened species, as well as brook floater and another state threatened species. And then yellow lamp mussel is the third state listed species. So it's a really high priority species in the state for us. So again, we're looking at life history. That's what I've been looking at. And really it's because this is a first step towards making informed conservation decisions about this species. So, freshwater mussels, right? I mean, why do we care? Well, really, it's because, well, they're more than just clams, right? I hear that a lot. They're just clams, but really, they have a lot going on, and they're really important parts of our rivers and streams, right? They're really essential parts of our ecosystem. So just a little bit of background. There's about 300 species that are native to North America, and about 74% of which are either threatened, endangered, or extinct, right? Or they have some level of imperilment, right? Which should be pretty alarming, right? Um, and that's alarming because of all these essential ecosystem services that they provide. They are filtering our water, structuring our food webs, right? They're feeding on algae and stuff like that at the base of our food chain. So they're helping to structure the food webs from the bottom up. They're an essential food source for things like muskrats. And then they also help stabilize the benthos or the river bottom. So there's all these services that they're providing in the ecosystem, right? So we really want to help maintain that. And then also, they're really good bioindicators, right? So mussels, they're extremely sensitive to adverse water quality, right? So when we're watching mussels decline, hopefully that's telling us something, right? That's indicating to us that something's not right. And these are going to be maybe the first things to go, right? So we should, it should be quite alarming that 74% of the mussels in the US have some level of imperilment, right? Um, and then lastly, mussels, this is maybe the most interesting thing about them actually, is that they're actually parasites to fish, right? And so this, again, this is kind of going into that bioindicator type thing. When mussels are declining, not only might that mean that we have some adverse water quality things going on, but it might also mean that we have problems with the fish in the ecosystem because of that codependence there. So again, so what's a host fish, right? And so I'm going to just briefly describe the mussel's life cycle, right? So they have this parasitic life cycle, right? So we have male and female mussels, right? Um, Males fertilize the females, and then they brood a larval form of the mussel that we call Glaucidia. So the Glaucidia, this larval form, they attach to host fishes, and they actually attach to the gills. So you can see kind of in the diagram there, they actually insist on the gills of fish. And then they spend about 2 to maybe 3 weeks attached to that fish. And in that time, they complete a true metamorphosis and they transform into the juvenile form, which you can see on the right there. Um, so again, these host fish, they're required as part of the life cycle of the mussel. So without the host fish, they're not going to complete their life cycle, right? Um, and then maybe, another really interesting thing is mussels, they actually have host specificity, right? So not only do they need fish, they actually need certain specific species of fish, right? And depending on the species of mussel, they're going to require different species of fish. Um, and so that information on that host use isn't known for about half of the species known to the US. So there's about 150 species of mussel that we don't actually know how they complete their life cycle, essentially, right? Because we don't know what host fish that they require to metamorphosize. So again, I just want to cover maybe some possible threats to freshwater mussels, right? So there's obviously many anthropogenic impacts, right? Sedimentation, excess nutrients, road salt, habitat alteration, and other point and non-point source pollution. I mean, these are all things that could affect mussels and things that have been connected to the loss of mussels. So these are all things in the watershed that, we should be cognizant of. Um, Also, kind of like I've indicated already, the loss of host fish, right? So that's a big thing. Aquatic invasive species, so things like zebra mussels can impair like the movement of mussels. They compete for food sources, like with zebra mussels also filtering bacteria and algae. And then another threat is actually just the lack of life history information, right? So there's a lot of things that we just don't currently know about mussels, and we need to know these things to make informed conservation decisions. So that's a huge threat and something that I hope to address here. So getting specifically into yellow lamp mussel and the research that I've done, again, this is a species that the life history is currently poorly defined, and we have very limited information on its host use. It's a species of greatest conservation need in New York, so we really need to know more about it to make sound conservation decisions for the species. We've seen populations declining in the upper Susquehanna. We've seen populations from the Unadilla, the main stem of the Susquehanna, as well as many other places that have just disappeared, actually. Um, and then from the other main watershed that it's present in New York, the Hudson River watershed, it's also declining. And there on the right, you can actually see its range map. So it ranges from Georgia all the way up to New Brunswick. But you can see this area in the Susquehanna, it's really a stronghold for the yellow lamp mussel, and it's concerning to see it declining, right? And so we're trying to get at some of this life history information. And so one of these things that I researched was what host fish is the yellow lamp mussel capable of metamorphosizing on. And so I used the mussel culture facility at SUNY Cobleskill that I helped to develop to answer some of those questions. So I got a cool little video there on the right. So that's the yellow lamp mussel, right? And that is its lure. And you can see it's actually moving it. And it's what it's doing is it's actually mimicking a small fish, which is really great. And what's interesting about that is that's a small clue at what it might be using for its host fishes. So this mussel is able to attract other fish in to actually strike that lure to then release that larval form of mussel. Um, and so that should tell us that it's probably carnivorous or piscivorous fish that the yellow lamp mussel is using. But again, we needed to confirm that. Nobody has done that yet. And so that was my first objective, right? Is identifying the hosts and the effectiveness of those hosts. But then I also wanted to assess those traits of those fish. Is the fish native or non-native? What family of fish does it belong to? What feeding guild does it belong to? Again, is it piscivorous, carnivorous, insectivorous, that kind of stuff? Um, what is the habitat preference of the fish? And does it overlap with the habitat preference of the yellow lamp mussel? And then we also, this is kind of extra, but we assess the timing of metamorphosis. So how long in the process of being encysted does it take to metamorphosize? And then we also determine the fecundity of female yellow lamp mussels. So just kind of getting into my study, I'm going to kind of work through the different components here. Um, so in order to test host fishes, we need to do this all in the lab, right? Um, so the first thing we did was we actually collected a number of different fish species. So we did this via boat and backpack electrofishing, as well as hook and line. And we did this from water bodies that we knew were devoid of yellow lamp mussel, um, just in case there is some gained immunity that would affect the metamorphosis rates that we observed. And then secondly, uh, we had to collect the female yellow lamp mussel brood stock. So we did that in from May to June, and we used 5 female yellow lamp mussels in our 4 trials. So 5 yellow lamp mussels in each of our 4 trials. So total of 20 mussels. And we collected most of them from Schoharie Creek, although we did collect some from the Butternut as well in the upper Susquehanna. So these are mostly yellow lamp mussel from the Mohawk River watershed, which is obviously part of the Hudson River watershed. So there's a lot going on here, but this is just a summary of my 4 trials. So you can see on the left there, trial 1, 2, 3, and 4. And so I think the big highlight here is I tested about 21 different species of fish, which is really huge. And we covered a number of different fish families, both native, non-native fish, a number of different diet preferences that you can see there, a number of different habitat preferences. Um, and really, this all culminates to um, it's going to be a really in-depth knowledge of the host use, right? It would have been short-sighted to just look at piscivorous fish when there's many other possibilities out there. So on the infestation day, we actually have to extract the Glaucidia from the female yellow lamp mussels. And so we did this using the syringe method. And so essentially we take a 3 milliliter syringe and we inject water into the mussel gills. And basically this flushes out any of the Glaucidia that are in the gills. And so you can see in the dish there, that's actually all that white stuff in the dish. Those are all little Glaucidia. So there's actually a ton of Glaucidia um, that the female yellow lamp mussels are able to produce. So then we did a salt exposure to test the viability of that Glaucidia. Um, and then we also enumerated the number of Glaucidia, and we did that volumetrically. And so we did that for each of the yellow lamp mussel brood stock. And we did this in order to find the fecundity of female yellow lamp mussels. And then lastly, so once we extracted all of that Glaucidia, we uh, made these infestation baths. You can see that there on the right. So it's actually just a 5-gallon bucket. Um, so all of those Glaucidia were put into the buckets, and then you aerate it really heavily, and then you put the host fish into that bucket, and they stay there for about 30 minutes before they move into the experimental system. So just real quick, this is the actual fecundity that I was talking about. So the average fecundity for the female yellow lamp mussel I found to be about 225,000 glaucidia per female, which is actually only moderate for mussels. It's only a moderate fecundity. Some species will have as many as 4 million. But again, this is still a lot. And then just a really quick summary of the viability and the infestation baths. We had a lot of viable glaucidia, and uh, we far exceeded the number of viable glaucidia per milliliter in our infestation baths that was targeted. So this is the experimental system on the right. We call these AHABs, or aquatic habitat units. So you can see there is actually 30 tanks in these systems, and we had two of these systems. So at any given time, we could have 60 infested fish. So each one of them being assigned their own individual tank. And then each one actually has these little PVC cups that are lined with a micron mesh. And these collect all of the glaucidia or juvenile drop-offs from each day. So in order to count every other day, we'd actually pull each one of those cups from the rack, and then we would take it to a dissecting scope. We would flush out any of the juveniles or glaucidia, and we would count each of those. And so this is what that could look like, right? So on the left there, it's a good representation of glaucidia, and on the right, you can actually see what the juveniles look like. And it's kind of obvious because they actually will stick their foot right out and well, and like move around. It's pretty cool. Um, and you can kind of see these are some total counts. So in total, I counted close to 300,000 glaucidia and over 10,000 juveniles. So I had a little counter as I was like going through the microscope, and oh, my thumb got sore on some days. It was pretty intense counting all of those. And so this is just broken out by an individual species, right? So this is creek chub, and so we have the number of glaucidia or juveniles there on the y-axis, and then along the x-axis, you can see that's by trial day. So days 0 through 25. You can see it's usually characterized by a high number of glaucidia dropped off early on in the infestation. Um, and then eventually you should hopefully have some juveniles drop off, but that usually doesn't happen until after day 15. And so spoiler alert, walleye worked really, really good. So you can see that there was a ton of juveniles that dropped off after day 15 through day 20. So that was really awesome. All right, so just getting into some of my statistical methods really quick. So all my models and plots, I did those in R in RStudio. And so for my host fish models, I use generalized linear models, or GLMs, with a response of juveniles, right? And then so I made 5 models for the different fish traits that included the species of fish, whether the fish was native or not, the family, the preferred habitat, as well as the feeding guild. And then I ranked those using AIC scores, so from best to worst. And then lastly, I assessed the timing of metamorphosis, and I did that again using GLMs, and I was assessing the probability of metamorphosis occurring over time. And I looked at time through the lens of accumulated thermal units, or maybe degree days, if you know what degree days are. So this is a really quick summary of the host fish models. Um, Basically, in short, there's really high significance across the board, and a lot of that is just due because I had about 4,000 lines of data informing these models, so they're all highly significant. Um, all of the R squares are like absolutely ridiculous as well. So really, this isn't actually telling us that much. It is telling us that species is the best indicator, but again, they're all pretty highly significant. So moving forward with species being that kind of best indicator there, this is all of our metamorphosis rates, right? So really what this is showing us is how good of a host are you? Um, all of my fish produce at least one juvenile, but they're metamorphosizing at different rates, right? So some are better than others. You can see walleye, smallmouth bass, black crappie, as well as brook trout were the four best hosts. Um, and that's a little interesting. You get back to some of those traits. Walleye, smallmouth bass, and black crappie, those are all non-native fish to the Atlantic slope drainages. So that's certainly interesting that those would be working the best, being that they're not actually native to here. You can also see that these are all piscivorous or carnivorous fish, which kind of aligns with what we would have thought. So next up, we have that timing of metamorphosis. And I just have this zoomed in on one species here. So again, on the x-axis, we have these accumulated thermal units, which is really just that it's a measure of time, but it's done um, through the lens of temperature. And then on the y-axis, we have the probability of metamorphosis occurring, right? So take that a step further. We actually see all of them for all the species that I tested. And well, really, this is just kind of confusing, right? There's a lot going on here, and it's kind of hard to tell what's going on. But as I started to look a little bit closer, some of our better hosts, like walleye or smallmouth bass, it kind of seemed like they are metamorphosizing a little sooner than a lot of the other hosts that didn't work as well. So I ended up grouping those as viable versus not viable hosts. And you can see there's actually this like pretty clear difference in the timing of metamorphosis. So if you're a viable host like walleye, smallmouth bass, black crappie, brook trout, you're actually metamorphosizing sooner than all of the other fish that we tested. And we need to look into this more, but it seems like there's some immune response going on there that with a stronger immune response, it actually takes longer for the mussel to metamorphosize. So there's some more research that needs to be done there and why that might be. Um, so something that somebody should look into a little bit more. So just some overall host trends. Again, so we have this piscivorous and carnivorous fish trend, which again, it makes some sense, right? And hopefully maybe this guides some additional fish that could be tested. There's certainly more fish out there than these 21 fish that I tested. Um, there's some species like longnose gar or chain pickerel that should still be tested, right? These are piscivorous fish that might be very good hosts, and we should learn some more still. There's also this weird thing with the non-native fish being pretty good hosts in this instance, right? Um, it's a little strange, right? And it brings up this question about migratory fish hosts, actually. It would seem that there was probably some historical importance with the striped bass, but due to habitat fragmentation with dams, obviously striped bass do not get to the upper Susquehanna watershed anymore, like they may have at one point. So their striped bass was likely the primary host for yellow Lampmussel at one point. But it seems that these non-native fish have probably taken that role as the primary hosts. And really, this is kind of good news in that just because they lost access to their primary host, they had these other fish actually take over that role. Obviously, the same can't be said for species of mussel like the eastern elliptio that requires American eel. It doesn't seem like another fish has taken over that role for them. So this is really good information to learn. What does this actually mean for conservation? This is actually just a really first really important first step towards determining the needs of each individual population. Are the hosts present? That's a really easy first question. Now, whether you can say yes, walleye are present, or no, they are not, this is a really easy place to divert your attention, right? So if they're there, great. That's not a problem, and we can go and look at some of these other um, possible problems, some of these anthropogenic issues that might be causing the declines, right? So we can kind of narrow it down what's actually going on that might be impacting this specific population. So this is a really nice box that we can check off. Um, with longevity and fecundity information, we can also assess the risk of future imperilment, right? So on a population level, we can look at the age structure that is currently there, and we can look at the fecundity and longevity and make a determination of how at risk is this population for imperilment, or are they doing just fine, right? Um, we can make more accurate assessments for imperilment status. So things like state species assessments, or say the U.S. Fish and Wildlife Service is considering listing the species. This is all information that they're going to need to use to make an accurate assessment for their imperilment status. And then lastly, this is, something I've been working on at SUNY Cobleskill with our propagation facility, is actually just maximizing hatchery production. So if we ever want to do some conservation aquaculture measures where we actually produce them in a hatchery setting and then stock them into the wild, now that we know what hosts work the best, we can maximize the production of the lamp mussel. And with that, I'd like to acknowledge my advisors. So I've been working with Paul H. Lord at SUNY Oneonta, Andrew Gasho-Landis at SUNY Cobleskill, as well as Dan Stitch, also at SUNY Oneonta. And then I'd also just like to acknowledge my funding contributions that I've received from SUNY Oneonta and their student grant program, as well as the Upper Susquehanna Conservation Alliance. And with that, I'll take any questions.
Great. Thanks, Brian. Great presentation. We only got a couple minutes left here, but if we need to run over, I'm sure you'll be available to answer some questions. We got
Yeah,
one in the
absolutely.
chat, and I don't know if this has been answered throughout the presentation. I think you might have talked about this a little more, but Adrian's asking if you could explain some more connectivity and dependency of mussels and fish in more detail, and their and how they interact.
Yeah, definitely. I mean, yeah, I definitely touched on it a little bit, but um, at this point, I mean, this is talking yellow lamp mussel specifically. It seems as like host fish shouldn't be a problem, right? In the upper Susquehanna, as well as many of the Atlantic slope drainages, smallmouth bass and walleye are pretty abundant. They've been spread widely. If you're into fishing, you go fishing. These are some of the most encountered fishes that you'll come across, right? So connectivity probably not a problem for the yellow lamp mussel. However, there is this historical angle that you can take at it, and that these fish weren't always around, right? And striped bass being a migratory fish migrating from the ocean, they probably played a huge role as a host historically. So there is a case to be made about um, dam removal, um, better connectivity, fish passage, that kind of stuff, even still, right? Um, it's not as nice of an argument, I guess, as something like the Eastern Elliptio, where it's pretty clear that they need American eel. Um, but even still, yeah.
I have one question, Brian.
Yeah,
The size, it seems like the size of the fish would be relevant in your study,
sure.
comparative to their gill surface area. How did you
Sure.
go about choosing the sizes of the fish that you used?
So really, as far as the size of the fish goes, it's whatever could actually fit in the in my system, the AHAB system, right? So I obviously was working with a limited tank size, so I needed to select a fish that could comfortably fit in those tanks. So the cool thing about metamorphosis rate though is that rate it's accommodating the size of the fish, right? So even though I'm working with a really tiny fish compared to some of the other fish that might be bigger and have more surface area on the gills, I'm that's all getting uh, translated to a rate that is comparable across the board, um, independent of the size of the fish. So,
Awesome. Well, thanks again for your presentation. Here's next week's presentation. It's going to be assessing road crossings in the Upper Tyapneoga River watershed by Cortland County Soil and Water Conservation District conservation assistants Jake Lynch and Brandon Ashmetat. So it's going to be our last session of the year, and we look forward to that next week. Thanks again, Brian, and thanks again for everyone for joining us this morning. And
yep,
enjoy the rest
thank
of your
you.
day.